Muy buenos días. Bienvenidos todos y todas a Frontiers in Systematics, Evolution and Biodiversity. Antes de empezar, un breve anuncio. Si suena la alarma del sismo o cualquier urgencia, la mitad de la sala sale por esta puerta y la otra mitad de la sala por la otra, por favor. Pues hoy la invitada será presentada por nuestra directora, la doctora Susana Magallón.
Muchas gracias, David. Antes de presentar a nuestra invitada del seminario Fronteras en Sistemática, Evolución y Biodiversidad, quiero hacer una mención especial de un artículo muy importante que fue publicado en la pasada semana. Es un artículo publicado por la doctora Gabriela Parra y sus colaboradores. Es un artículo que se llama Ongoing Declines for the World's Amphibians in the Face of Emerging Threats, publicado en la revista Nature. Y quisiera destacar, bueno, nosotros sabemos que las revisiones sistemáticas son indispensables para la implementación de acciones de conservación y por lo tanto la disponibilidad de nuevos avances en taxonomía y sobre todo de los entendimientos de las causas que conducen a la extinción hacen que una revisión, las revisiones frecuentes sean imprescindibles. El artículo que fue publicado es el reporte de la segunda revisión global de la diversidad de anfibios, basada en una evaluación de más de 8,000 especies para la lista roja de especies amenazadas de la Unión Internacional para la Conservación de la Naturaleza, la IUCN. Y los hallazgos son pues muy importantes. Encontraron que los anfibios son el tipo de vertebrados más amenazados, ya que alrededor del 40% de las especies a nivel mundial se consideran amenazadas. Y sobre todo muestran especial preocupación para las salamandras del neotrópico, en gran parte debido a la pérdida del hábitat y de las enfermedades que sufren. También se enfatiza la necesidad de llevar a cabo acciones inmediatas para detener las tendencias observadas. Entonces, si bien los resultados de este artículo nos ponen en una gran alerta, quiero hacer una gran felicitación a la doctora Parra y sus colaboradores por la publicación de este artículo. Ahora sí, me da un muy gran gusto presentar a la doctora Cory Moreau, quien es nuestra ponente en esta serie de seminarios de fronteras en sistemática, evolución y biodiversidad. Ella iba a dar su charla en línea en mayo, pero pensamos que es mucho más agradable que nos viniera a visitar. Entonces tenemos el gusto de tenerla aquí presencialmente. La doctora Moreau es decana en temas de diversidad, equidad e inclusión de la Escuela de Agricultura y Ciencias de la Vida de la Universidad de Cornell en Nueva York. También ella es profesora titular de la cátedra Martha y John Moser en Biosistemática y Biodiversidad de Artrópodos en el Departamento de Ecología y Biología Evolutiva de la Universidad de Cornell. Y también ella es directora y curadora científica de la colección de insectos de la Universidad de Cornell, en la cual tienen más de 7 millones de especímenes en su acervo. Ella realizó sus estudios de licenciatura y maestría en la Universidad Estatal de San Francisco y el doctorado en biología evolutiva en la Universidad de Harvard. Y también fue un Miller Fellow en la Universidad de California en Berkeley. Ella ha recibido múltiples distinciones. Por ejemplo, en 2014 fue exploradora de National Geographic. En 2018 también fue considerada como una mujer de impacto también por la National Geographical Society. En 2016 fue miembro cabil de la Academia Nacional de Ciencias de los Estados Unidos. En el 2018 fue miembro de la Academia Americana para el Avance de la Ciencia. En 2020 fue nombrada miembro de la Sociedad Entomológica de los Estados Unidos y en el 2021 fue nombrada miembro de la Sociedad Real de Entomología. Ella ha recibido más bien un género y 3 especies han sido nombradas en honor de la doctora Moreau. En sus investigaciones, ella ha publicado más de 100 artículos científicos y sus investigaciones están dedicadas a la evolución y la diversificación de hormigas junto con sus bacterias simbióticas, combinando trabajo de campo con herramientas moleculares y genómicas para resolver preguntas de cómo las especies participantes en sistemas evolutivos se benefician de estas interacciones simbióticas. También ella tiene interés en el papel de la biogeografía y la evolución de caracteres y la simbiosis en los procesos macroevolutivos con el objeto de comprender los patrones a gran escala de la evolución biótica. También ella tiene gran interés en promover la comunicación pública de la ciencia e incrementar la participación de una diversidad de personas en la ciencia. Y sin más, le doy la bienvenida a la doctora Moreau, quien nos va a presentar la plática titulada Macroevolution to Microbiomes, Piecing Together the Puzzle to Understand the Evolution of Moths. Welcome, Cory. Thank you.
Thank you so much all for that could attend in person and online. It is a great honor for me to be here. I have always wanted to visit UNAM. You know, there's those institutions around the world that you've known many people to come through and go on to successful careers, or you've known the faculty and their publication records. And so for me, this was a place that I'd always wanted to visit. So I'm so excited to be here. I'm so excited to meet all of you. I've had such wonderful conversations. I've seen some of your beautiful collections. I've heard about the fantastic work you're doing, so I'm just very pleased to be here. So what I'm really fascinated with is sort of when I go outside, the biodiversity I see. And to me, what I really think about are what are the processes that have generated that biodiversity? I think about that in the context of both the abiotic and the biotic factors. From the abiotic factors, I might think about things like temperature, precipitation, soil quality. Thinking about the biotic factors, I might think about things like predator-prey interactions or symbiotic interactions that are helping facilitate that biodiversity that we see. And ultimately, what I want to try to understand is how do these sort of interspecies interactions really drive the ecology and evolution of the species we see on the planet today? As an evolutionary biologist, I see the lens entirely through the lens of a phylogeny. When I look at species, I want to know how they're related. Now, depending on what group of organisms you're interested in, the phylogeny itself might be more or less interesting to you. As an entomologist, this phylogeny is not very interesting to me. But what I understand about a phylogeny is the power of what it can tell us. It can tell us who's more closely related to who, right? In this case, you might want to understand who's the sister taxa to the chimpanzees. But the power of a phylogeny is what you can use it to infer. So in this case, maybe what we want to understand is the evolution of traits. How many times did spines evolve? How many times did we see these intimate interactions evolve? Are those due to a single evolutionary event, or is it due to convergence? And the kinds of traits we can look at are, of course, anatomical and ecological and behavioral, but we can also think about the evolution of traits like where they're found on the planet. We can begin to ask questions about biogeography. How did those species sort of expand and migrate out of the place where they evolved? We might also want to ask questions about speciation events. Why did something that was one species diverge into two? Can we correlate that with something we know about the geology or geography of the area? Was it one sort of large population and then a mountain range arose and divided them? Did a river change course and that now explains why they're split? And maybe that sort of divergence has just happened through time. We may also want to understand something about, rates of change. So almost all of my work is based on molecules, so DNA and genomics. So I am often asking questions about rates of molecular change. Why is it that some parts of the tree of life have really fast rates of molecular evolution while others have slow? But of course, we can ask that through, questions about morphology. Why is it that some lineages have fast rates of morphological evolution while others, being much older, have really slow rates of morphological change? And if we have some kind of external information, whether it's the fossil record or maybe major geologic events, we can put a time frame on that. We can ask, when did those traits evolve? When did those speciation events occur? So one of my PhD advisors was E.O. Wilson, and he once said that ants and other insects are the little things that run the world. And of course, being an ant biologist, I completely believe this. But I think one of the things that makes ants such a powerful system to ask questions in biology is that they engage in symbioses across the entire tree of life. We know that they engage in symbiotic interactions with lots of kinds of animals. In this case, on the left, you see this ant tending a scale insect. And in this case, the scale insect will produce this droplet of honeydew that the ant is holding in her mouth. And in return, the ants will defend that scale insect from any parasites, predators, parasitoids, right? So there's a sort of symbiotic interaction happening there. We also know that they engage in symbiotic interactions with lots of kinds of plants. The plant you see here is, I call it little A acacia. It's now called Vachelia. You guys have them here in Mexico. And this is sort of the quintessential example of an ant-plant interaction. And I'll talk about some of our work there with the acacia, but of course they interact with lots of other kinds of plants. The most classic example of a symbiosis in ants I tend to think of are things like the fungus farming ants, right? Where they're cutting down pieces of vegetation to grow their fungal colonies. But not all symbiotic interactions are positive. In this case, you see an ant that succumbed to the Orthocordyceps, or the zombie fungus. And I've done some work with David Hughes's lab trying to understand the evolution of this zombie ant biogeography. Now, the more and more that we study ants, we understand that these interactions even manifest in their microbial interactions through things like their gut microbiome. And so I like to say now that the microbes inside the ants are the little things that run the little things that run the world. So what I'll do is I'll walk you through two broad research programs we have in my lab. One is asking questions about the macroevolution of ants themselves, and the other is trying to understand the microbiome. And so first, I'd like to walk you through a story in which we tried to understand, did ants and plants that are interacting through evolutionary time have sort of an increase in interdependence? So anyone who studies flowering plants, and I already see Susanna smiling, you know that in the past, there was a lot of diversity of plants on the planet, but really right now we're in the age of angiosperms, or the flowering plants, right? They take up the majority of plants we see anywhere in the world. And as those flowering plant forests diversified, many other groups did as well. Many kinds of insects that either pollinate those flowering plants or feed on those flowering plants went through bursts in diversification. In addition, lineages you might not think as clearly would have an association, things like epiphytes and ferns also went through bursts in diversification. And it's thought that the low light created by this dense canopy allowed them to flourish. So it probably isn't surprising to know that many groups went through bursts in diversification, and some work that I did earlier in my career demonstrated that ants also went through a burst in diversification as the flowering plants expanded across the globe. So one of the things that we also know is that as ants expanded into these flowering plant forests, their food resources diversified. They began to feed a lot more on plants, either directly through things like extra flow nectaries, the food bodies that are produced by plants to attract ants, or even things like elaiosomes. So these are these little fatty bodies that are produced by plants on their seeds to attract ants to come and drag the seed away from the parental plant. So you no longer have competition between the parental plant and their offspring. In addition, I would say a lot of the diet of ants that are feeding on plants is actually indirectly, and that's because they tend sap-sucking insects, like the scale insect we saw previously, where the sap-sucking insects will produce honeydew to attract ants. They'll feed on this almost unchanged plant-derived resource and then defend those insects in return. So what we wanted to understand is sort of how did these relationships evolve? Are the ages of when we see these, behaviors evolving in ants and when we see these traits evolving in plants, are they correlated at all? And is there some evolutionary sequence that makes sense that could explain the history of this? So the first thing we did is we capitalized on a really large plant phylogeny that had almost 11,000 plant genera in it. We actually augmented it by adding in a few more fern lineages, because it turns out that ferns also have associations with ants. And then we did some ancestral state reconstruction on characters in plants that are associated with their interactions with ants. So we looked at things like extrafloral nectaries, whether or not they've evolved domatia, the specialized structure to house the ants, and whether they have lyosomes. And on the ant side, we inferred the largest data-driven ant phylogeny to date. It has over 1,700 species in the phylogeny. And then again, we did ancestral state reconstruction on things related to their diet, their nesting, their associations with plants. Now, this was work that was really spearheaded by a former postdoc in my lab, Matt Nelson. So before I sort of walk you through the results, I want to remind you that the earliest ants were predatory. They were nesting in the ground. They were foraging on the ground looking for food. And so what we see in this sort of sequence of ant-plant evolution is the first thing we see is that ants leave the ground to start to forage arborally, right? They're looking for prey items up in this flowering plant canopy. Next, we see that ants begin to include plants in their diet. Initially, it was probably through things like plant wounds, and there's been some beautiful work done by some colleagues that have now demonstrated that ants are highly attracted to plant wounds, and that may be the likely transition into feeding more directly on plants. Now we see the plants begin to respond. So there's this, large number of ants roaming all over these plants. And we see plants now have evolved extrafloral nectaries, right? So they're taking advantage of this army of ants to attract them to some plants so that they'll act as deterrents for herbivores. Now we see that ants move up into the canopy and are building their nests there. And we see that the plants now again respond and evolve lyosomes. So they're taking advantage that ants are just everywhere, and they're capitalizing on the fact that they can leverage them to provide services for them. And lastly, we see that most complicated structure that has no other use but to house defensive arthropods evolve, the evolution of domatia. So what this really allowed us to see was that the ants were utilizing plants long before the plants evolved these specialized structures. And that what we see is this sort of incremental sequence of evolution that had led to the diversity we see today. And so what we see is that ants transition from being predatory, ground nesting, and ground foraging to beginning to forage arborally. They begin to include plants in their diet, and eventually they begin to nest arborally. Now the plants begin to respond by evolving specialized structures just to attract ants. First, they evolved extrafloral nectaries, then they evolved lyosomes, and then finally domatia. Now, one thing we hadn't set out to test when we did this study, but we were able to address, was this long-standing hypothesis about diet shifts within ants. It had long been sort of known in the ant community that although the ancestral condition for ants were that they were predatory, that once you became herbivorous, you never reversed. Well, we actually were able to test that. And what we showed is that in order to become herbivorous, you always had to transition through being a generalist or having an omnivorous diet. And once you evolve to herbivory, strict herbivory, we never see a reversal back to being predatory, and almost no reversals back to even being omnivorous. So it appears that once you're locked into this plant diet, it really is an open niche for ants to be able to diversify. So hopefully I've convinced you that ants and plants have this really rich, long evolutionary history. And so what I'll dive into now is some work we've done specifically on ant-plant mutualisms and asking the question, how do we expect the genomes of these ants to sort of respond to this relationship? One of the big questions in sort of symbiotic literature is, why do mutualisms evolve and why do they persist through time? Some people have hypothesized that these are evolutionary dead ends, right? Once your relationship with another organism becomes so intimately entwined, if one or other of you has any kind of drop in your populations or goes extinct, both will crash. And so it begs the question, why do we see these relationships evolve? I really love this quote by Richard Spruce from the 1800s when he was asked about ant-plant relationships. And he said, plants need ants like dogs need fleas. So he could see how the ants were benefiting, but he thought that the plants were just being taken advantage of. We now know that, of course, that's not true. And I really love this paper by Marjorie Weber and Anurag Agarwal, where they investigated the evolution of these defensive mutualisms across a large number of flowering plants. And what they showed is that when plants engage in these mutualistic interactions with defensive arthropods, ants and mites, we see not only increased rates of speciation, we see increased rates of diversification in those plant lineages that engage in this, suggesting that it is beneficial for the plants. So everyone has a different definition of symbiosis, so I'll share mine. For me, symbiosis is the close and often long-term relationships between two or more species. And in this case, what we wanted to ask is, are there predictions about how it should drive genome evolution? Now, if you think about it, there's sort of a well-known example, and this was a hypothesis put forward by Lee Van Valen in 1973, where he gave an homage to Lewis Carroll's Through the Looking Glass, right? And this is called the Red Queen hypothesis, where he uses the analogy of when Alice is talking to the Red Queen and they're, sort of running together. And the Red Queen says, it takes all the running you can do to keep in the same place. And this is the idea of like an evolutionary arms race, right? We've all heard of this. You can think about something like where a parasite evolves to outwit the immune system of the host, then the host of immune system evolves again to outwit the parasite, and so on and so forth. So we see this increased rate of evolution, in this case, even playing out at the molecular level. And so in these examples, it's been well documented that in these parasitic or antagonistic interactions, we see increased rates of molecular evolution, specifically at the genes that are involved in that, things like immunity, but it can play out in other parts of the genome. Now, the best way to study these kinds of interactions is through sort of, bacteria in plates. And one of the trouble we have is when we ask questions about mutualisms, when you start to evolve a mutualist, a microbe in a plate, they start throwing out whole chunks of their genome so that their genomes reduce rapidly. So making direct comparisons becomes more difficult. But there has been a body of theoretical and mathematical literature that's made predictions about what we should expect. And this is called the Red King effect. And in this case, the hypothesis is that when you have these mutualisms evolve, it's predicted we'll see a decrease in the rate of molecular evolution by decreasing the fitness of uncommon genotypes and therefore reducing the rate of molecular evolution. And that can happen in one or both partners. So what we wanted to do was to set out and test this question. So we looked to this genus of ants, Pseudomyrmex, which commonly include the acacia ants. And in this case, some of the species are mutualistic with these plants. So in this case, they're living inside those specialized domatia where they store their food and rear their young. And they live exclusively on their tree. They get all of their food sources from the host plant through things like extrafloral nectaries and these Beltian or food bodies. Now, in return, the ants will aggressively defend their host plant from any herbivore that comes nearby. In fact, if you even walk close to the tree, the ants will jump off and sting you. And it's not just herbivores they're keeping away. They actually also will climb down to the ground and trim any encroaching vegetation, so there's no competition with their host plant for resources. And they glean pathogenic fungi off of all of the leaf surfaces. So in this case, really the plant has outsourced its defense to its ant partner. Now, within the genus Pseudomyrmex, about 40 species are mutualists. And in this case, they all have identical behaviors. They're all defending their host plant. They're all patrolling their plants 24 hours a day, and they're highly aggressive and have really large colonies. But interestingly, their relationship with plants, although it's one-to-one, one species of Pseudomyrmex is only found with one species of plant. Interestingly, the plants that they partner with come from 3 different genera and 2 different plant subfamilies. But the majority of species in the genus are actually generalist with a couple of species that are parasites. And in this case, they nest opportunistically in any hollow twig they can find. Now, because they're not aggressive, they have a very different behavior. So if their nest is bothered, they immediately flee. They don't even protect their own young. So with my former graduate student, Ben Rubin, we set out to let evolution play out the experiment for us so we could ask the question, what happens to the genome when you become intimately involved in these obligate symbiotic relationships? So within the genus Pseudomyrmex, this relationship with being a mutualist with a group of plants has evolved 3 times independently. So when we started this project, there were actually no published ant genomes. We knew that there were some in the pipeline, and by the time we were getting ready to publish our results, 8 ant genomes had been sequenced. We, of course, compared all of our results against those 8 genomes, but bear in mind that those genomes are separated from Pseudomyrmex by probably about 75 million years. So it's not very close related, so it didn't really leave us with much genomic resources. So in order to address this question, the first thing we had to do was to sequence a reference genome. So we used an outgroup from within the genus Pseudomyrmex gracilis to do this. Now, one of the beauties of working with Hymenoptera is that they're haplodiploid. For those of you who are not insect biologists, I'll remind you that in this case, when a queen lays an egg and unites it with sperm, it becomes diploid and becomes female. When she lays an egg and does not unite it with sperm, it becomes male and it's haploid. So we could lever this sex reproduction strategy by sequencing the genome of a single male that's haploid, which makes assembly of the genome much more straightforward. We then took all the rest of the life stages, the queens, the workers, the larvae, the eggs, ground them up and sequenced their transcriptome so that we could then annotate our genome very well. And by all metrics that you can use, we have a very good reference genome to be able to do the remaining analyses. We then went one step further and did some genotyping by sequencing, and we're able to recover 38 linkage groups for this genome. Now, this species is known to have 36 chromosomes, so we're pretty close to having a incredibly well assembled, annotated, and almost chromosome level genome to be able to do our comparisons. We then went in and sequenced the remaining 6 genomes and mapped them back to the reference genome. Now, we found 2 sort of striking findings that I'm going to share with you right away, and then I'll walk you through one that I think is particularly interesting. So what I told you about these ants is the thing that makes the mutualists different is their behavior. They look almost identical to their generalist relatives. And so what we found is incredibly strong positive selection on genes related to nervous system. And so neurogenesis and other nervous system related genes. And that actually makes sense then, since what's different is their behavior. Now, we used an incredibly conservative approach. We only considered genes that were under positive selection in all 3 mutualists and not under selection under the 3 generalists. So clearly we missed a lot of genes that are probably responsible for the species level mutualistic behaviors. The other thing that we found was that in all of the mutualist genomes, we found faster rates of molecular evolution, which is exactly against that body of theoretical literature, suggesting that something different is happening here. Sorry, this is a transition from Mac to Windows. I'm just going to skip this slide because it's sort of more clearly shown in the next slide. So what we were able to do is to take those pairs of the mutualist and the generalist that are most closely related in each of those 3 and compare them to the outgroup. That's what the last slide would have showed you. And then we did sliding windows across the entire genome to measure, do we see faster rates in the mutualists or the generalists? And in all cases, overall, we see faster rates in the mutualists. So that's true whether we look at the genome or whether we look at the coding components of the genome. So in this case, what we see is just faster rates of molecular evolution across the genome. And this holds for DS and in 2 of the 3 cases of DN where it's not significantly different, suggesting something really is happening in these mutualistic genomes. Now, of course, there's lots of demographic reasons we might be seeing this pattern, so we wanted to test those. The first could be related to generation time. Maybe what we see is that the mutualists just have sort of slower generation times than those generalists. So the way I like to think about it is, let's imagine that we decided right now as a group of biologists, we're like, let's conduct an experimental evolutionary study. We're going to let it run for 100 years. We're going to take an elephant and we're going to take a mouse and we're going to watch how many mutations accumulate in their populations. Well, in 100 years, those mice have just reproduced, and even without any selection, just due to genetic drift, we would see more mutations in the mice that ended 100 years. Where the elephant, we might look at the same individual, which means no mutations accumulate, or maybe one offspring, so we don't have time. So maybe what we have is that these mutualists just have a slower, a rate of generation times. But if you remember, I told you in the beginning that they have very different ways in which they build up their colonies and defend themselves. Since the generalists have to go into reproductive mode almost right away, usually within about 2 months, they're already producing sexuals because they don't defend their colony. That's completely different than the mutualists that have to build up this standing army of workers to defend their host tree before they can invest in reproduction. So what we know is that it usually takes about 1.5 to 2 years for the mutualists to actually go in reproductive mode. So in this case, we can actually rule out generation time. Now, the next thing could just be population size. Maybe what we see is that the mutualists just have smaller population sizes, despite the fact if you go to a location that has these mutualists, they're in very high density compared to all their congener generalists. So in order to test this, we took a 2-pronged approach. We looked at the ratio of DN to DS across all the genomes, and we also used the genomes themselves to estimate population size, because unfortunately, no one has done the careful population genetic studies for all these species. And in both situations, what we see is that we can rule out population size. We don't seem to see that these mutualists have smaller population sizes. Now, it could just be we're seeing the signature of relaxed selection happening in these mutualists. And in order to look at this, we looked at the level of repetitiveness we see across the genome. And what we see is that it's not consistently different. So it doesn't seem to be the smoking gun that explains these differences, although we can't necessarily rule it out. And last, maybe we just have faster elevated mutation rates in these mutualists. So to look at this, we looked at the heterozygosity and the differences between these genomes. And what we see is we do see a trend toward higher rates within the mutualists. Now, I should share that this is where this paper ended. We tried to speculate as to why we thought this might be, and that maybe it's this idea of the red queen, constantly running to stay in the same place, and that obligate mutualisms should also be held within the window of just any kind of a symbiotic relationship like we see in parasites. But if you hire smart students, which I'm sure all of you in this room are, when that thing is tickling in the back of your head, you can't let it go. So my former graduate student had gone off and was doing a postdoc, and it was just bothering him. So on the side, he decided to pursue this further. What he did is he went in and used colony size as a proxy for both the size of a colony, but more likely the reproductive rate of the queens within both bees and in ants, and looked across multiple lineages. And interestingly, what he found were we see higher rates in species that have larger colony sizes. Now, this actually makes sense because we know for species that with age, you see more accumulation of mutations in your, oh my gosh, why am I going to forget the word right this moment? In your eggs and in your sperm, right? So I can't remember the word at this moment. And so really what we're likely seeing is that these older queens have more mutations accumulating in their reproductive cells. And so that's what's leading to this signature of elevated mutation rates. So if I had, another 30 years of my career ahead where I could do this very careful study, I'd take lots of species of ants, both that are generalists and mutualists. I'd sample the workers as soon as they're born in the colonies and as they age. And I predict what we'd see is that species that have really large colony sizes and live 25, 30 years, the workers are likely accumulating more mutations than those that were born early in the colony's life cycle. If any of you want to take that on as a project, please go right ahead. All right, so hopefully I've been able to convince you that when you look at the impact of things like mutualistic relationships or symbiotic relationships on genomes, we can actually see a whole variety of ways that they could impact genome evolution. And so really we have to think carefully about what it is we're actually testing. And in this case, you saw a scenario in which we couldn't even get to the answer until later after we'd published the paper. So hopefully I've convinced you that ants and plants have a very intimate relationship. And one of the things that really intrigued me as I went through my career was thinking about how these predatory ground nesting ants transition to feeding exclusively on plant-based diets. And that's really what led me down this path of asking questions about the microbiome. And specifically, we wanted to understand, did symbiotic bacteria allow ants to transition onto these herbivorous life cycles? So why do I think ants are such a great system to ask questions about the microbiome? Well, one of the reasons is that we know that, we can ask questions about diet and nutrition. So since ants are social species, we can take colonies, we can split them, and then we can actually manipulate their diets in real time and then watch how the microbiome responds. In addition, we can ask questions on an evolutionary scale because ants have evolved herbivory independently, probably at least 13 times. We can ask the question, what happens every time they evolve herbivory? We can ask questions about transmission. We can take a colony and introduce a pathogen and see what happens as it spreads through the colony. Are there hubs that are more likely to sort of pass that microbe around? Are there dead ends that protect the colony? We can also ask questions on an evolutionary scale about transmission. And that sort of leads into when do we expect to see sort of coevolution of host and symbiont? Why are there reasons we would expect it not to co-evolve and other situations in which we would expect it to co-evolve? And then lastly, because bacteria are everywhere and ants are everywhere except Antarctica, we can ask the question, are they just locally acquiring them from their environment? Or is there some other filtering mechanism that's determining what bacteria get into the guts of these ants? So I always joke that when we started this work, very little was known about the ant gut microbiome. So I always say that my colleague Jacob Russell, who's at Drexel University in Philadelphia, and I were sort of like those early rainforest explorers. We were just documenting what was there. We were grabbing every new thing we saw and putting it in our little collecting bag, right? And so when we started the study, we decided to sample almost 400 individual ants from almost 150 ant genera, carefully dissecting their guts and just saying, what do we find? And of course, what we found was a lot of diversity, not surprising. But what really jumped out to us was that we saw an enrichment in this group of bacteria, the Rhizobiales. Now, some of you who work on plants might be familiar with this group of bacteria, right? Because this order of bacteria includes the Rhizobiales, the ones at the top, right? So this group of bacteria have long been known to be associated with the roots of leguminous plants. So in this case, what happens is that the plant builds this little structure around the bacteria, right? And then the bacteria is taking advantage of the carbon resources produced by the plant. And in return, it's fixing atmospheric nitrogen and providing it to the host plant. So we wanted to understand, why are we finding a group of bacteria that are related, not super closely related, but related within the guts of ants? Could they be improving their nutrition in essentially the same way that they are plants? Another clue that we had that they might be facilitating the nutrition of the plants is that we use stable isotopes to infer the trophic ecology or the food web of ants. So what you're looking at is the enrichment of nitrogen as it moves up the food chain, with things like army ants all the way out on the right, and then things that are entirely herbivorous on the left. So each of those diamonds is an ant genus in which we've inferred the trophic ecology, so looked at the ratio of that heavy to light nitrogen in the habitats from which they're found. And then we've mapped on the y-axis whether we see association with this group of bacteria. And what you'll see is that almost all ants who are herbivorous are associated with this group of this order of bacteria. Now, if you have a keen eye, you likely notice that there's two dots that are feeding low on the trophic scale, but have no association with Rhizobiales. And that turns out to be one of the only ant groups in which we knew anything about their gut bacteria. And that's the carpenter ants and their relatives. And in this case, we already know that they have bacteria that have been co-associated with their hosts for a very long time evolutionarily and have a specialized structure called a bacteriocytes to hold the bacteria, and that they're upregulating their host's nutrition and are maternally transmitted. So it appears if you feed low on the trophic scale, you need an association with some kind of bacteria. So what you're looking at here is a phylogeny of the bacteria, color-coded and labeled by the host or the environment from which it came. And what you'll notice is that almost all of the tips in red come from a single clade, and they're all associated with ants. And we call that the Rhizobiales specific clade. Now, what's interesting is not just that most of the ant samples group together, even within that clade, they seem to be tracking the relationships of their hosts. So all of the samples from Cephalotes group together. Cephalotes is sister to Procryptoceras. So the bacteria within the guts of Procryptoceras and Cephalotes are each other's closest relatives, right? So we're seeing sort of what looks like a long-term relationship of this group of bacteria with their ant hosts. But if we flip it on its head and look at it through the lens of the ant, what we see is there's been at least 5 independent associations of this order of bacteria with their ant hosts. And interestingly, when we do some statistical analyses, it's highly correlated with ants that are herbivorous and that are living in the canopy, which has been hypothesized to essentially be a food desert, a place where there's not a lot of resources available, suggesting that they may really be relying on these gut bacteria to enrich their diets. Now, if these bacteria are in fact improving the nutrition of the host, we'd expect to see some amount of tissue specificity across the digestive tract. So just to remind you about ant internal anatomy, what we have is the first chamber of the digestive tract is called the crop. Now, no digestion happens in the crop. This is the social stomach. It's just where they can hold liquid resources so they can regurgitate it to other members of the colony as they come into contact with them. It's just like the crop in an adult bird, where they carry food back to regurgitate to their young. So no digestion occurs there. Digestion begins once we get into the midgut and the hindgut. In the hindgut, we further broke it out and dissected out the independent components of the hindgut. We looked at the ileum, which is essentially the large intestine, and the rectum, where we see excretion occurring. Now, after carefully dissecting each and every one of those compartments across the turtle ants, so many, many species, this is what we find. And this is some work that was spearheaded by a former graduate student in my lab, Peter Flynn. So each one of those bars is an individual tissue sample from a different species of turtle ant. And what you'll notice right away is the crop, or the social stomach, is really diverse. Every sample looks different from one another, but almost every sample is a single color, suggesting that it's really dominated by one group of bacteria. Now, once we move into the midgut, where digestion begins, almost all the samples are dominated by a single group of bacteria. This is the Opotutales. And then interestingly, once we get to the ileum and the rectum, we're recapitulating the rest of the known core bacteria that we find within the digestive tract. And in fact, the rectum really seems to just sort of, be influenced by everything that's upstream. Now, I have sat and done thousands of careful dissections on ant guts. What we wanted to know is, can we just pull off the abdomen or the gaster and sequence it so that we don't have to do the careful dissections? And then, in fact, what we see is we can, which is great news for future studies. But what's interesting is it's not just the diversity that we see that's structured across these gut compartments that suggests that they're functionally important. It's also the quantity. So using quantitative PCR, what you'll notice is that all those samples that come from the gut, although there was a lot of color variation among the samples, there's almost nothing there. It likely just reflects whatever contaminant that they picked up last when they were eating. And so it suggests that those bacteria within the crop are not functional, they're not diverse. Once we move into the midgut, we see a huge increase in the quantity of bacteria, dominated almost exclusively by one group of bacteria, the Opotutales. We see that the quantity of bacteria is maintained once we get to the ileum, where we know digestion also occurs, and then again drops off once we get to the rectum, where it's likely just involved in excretion. Now, I've been talking about ant-bacterial interactions, but as you just saw on the last slide, there must be bacterial interactions. So a former graduate student, Anaïs Chanson, wanted to investigate this. So bacteria have lots of ways of interacting both competitively and in cooperation with other bacteria. This is why we get all of our antibiotics from bacteria, right? And so we're going to ignore the crop and the gaster for a moment and only focus on those gut compartments in which we saw high amounts of quantity of bacteria. And what she did is she went in and looked at the genomes and metagenomes of the bacteria from these compartments and asked the question, how many have retained the biosynthetic gene clusters within their genomes? So within a bacterial genome, a biosynthetic gene cluster is so that it can produce antibiotics so that it can go to war with other bacteria. And interestingly, when we looked across all these genomes, we only found biosynthetic gene clusters in those bacteria that are found within the ileum and the rectum. So in the midgut, where we only have one bacteria, it's lost all the biosynthetic gene clusters because it's not in competition with other bacteria. But what we see is that there still must be some competition for resources going on in the ileum. And I'll walk you through why that might be in a moment. Now, interestingly, because we're seeing these diverse communities, we wanted to understand, do we see some of these bacterial communities co-diversifying or not? So when we sort of take the bird's eye view, so sort of step back, and in this case, we're looking at the families of bacteria, or in some cases, we're looking at single genera, we see that some show patterns of co-diversification while others do not. And some of it makes sense. Those bacteria that are alone in the midgut are co-diversifying. Things like Wolbachia are not, and that's exactly what we'd predict. And when we dive into one of the core members of that midgut, where it's all alone in the midgut, we see almost complete congruence of co-diversification of the host's phylogeny in this group of bacteria, suggesting that they've had a long evolutionary history together. Now, I told you that we found that they were related to a group of bacteria, the Rhizobiales, where we know that within the leguminous plants, they're fixing atmospheric nitrogen. So we wondered, could this be how the bacteria are enriching their host nutrition? So we took a two-pronged approach. We looked across the genomes for NIF genes. So NIF genes within a bacteria are there to fix atmospheric nitrogen. And what we did not find is functional NIF genes. We took one more approach where we did acetylene reduction assays to see, could we see the fixation of atmospheric nitrogen in either individual ants, groups of ants, different species of turtle ants. And in all cases, we see no signature of the fixation of atmospheric nitrogen. So if the bacteria are in fact enriching the host's diet, it's not the same way as we see in plants. Now, we do have other indirect evidence that they might be, and this comes back to the question about competition within the ileum. So what you're looking at here is a fluorescent in situ hybridization image of the gut of a turtle ant. The social stomach is just off screen. You next see that big round bulb is the midgut. Then you see the long intestines or the ileum, and then finally you see the rectum. It's a little destroyed on the end, but you can see the rectal pads clearly. And then those spaghetti-like structures are called the Malpighian tubules. And for those of you who are not up on your insect physiology, these are essentially the kidneys of an insect. They're floating in the body cavity and they're taking all the waste and depositing it within the digestive tract so that it can be excreted. And those Malpighian tubules connect to the digestive tract right at the juncture between the midgut and the ileum. And so what you're looking at is a stain here where it shows us the densest bacterial communities, and they're right at that juncture where the host body is depositing all that uric acid, the uric waste, right into the digestive tract. And that waste has been demonstrated can serve as the building block for bacteria to synthesize amino acids. So you could imagine that this is prime real estate for a bacteria. So that might explain why we see those bacterial competitions or the retention of those genes so they can fight for the best habitat within the digestive tract. Now, we do have a few direct lines of evidence that demonstrate that these bacteria are in fact enriching their host's diet. So along with my long-term collaborator, Jake Russell, my long-term collaborator, Yi Hu, and John Sanders, we've gone in and when we can culture these bacteria from the guts of the turtle ants, which is actually quite difficult, we sequence the genome of those. When we can't culture them and sequence an isolated genome, we've done a lot of metagenomic sequencing. So we've now been able to recapitulate all the bacteria within the guts. And what we've been able to demonstrate is all those core bacteria that we find always associated with turtle ants across their 50 million years of evolution, we see that they retain the genes to synthesize amino acids for the host. And in particular, they retain all the genes to synthesize the essential amino acids that are missing from animal diets. Now, as I've already told you, the bacteria are interacting with each other. So maybe they're synthesizing these amino acids as what's called a common good. It's I'll scratch your back if you'll scratch mine, right? So I'll produce these so that you give me some resource that I need in return. So we needed to demonstrate that the bacteria were synthesizing these and then they were being utilized by the host. So we didn't ask the question, are they present in the gut? Because again, those could just be for that common good relationship. So what we did is we actually went in and did a manipulative experiment. We took multiple species of these turtle ants, divided them in half and gave half antibiotics and half not, so we could knock out or knock down their bacterial communities. We then fed some of them an enriched form of urea in their diets. And then we asked the question, do we see that urea, do we see those essential amino acids, not in the gut of the host, but translated all the way into the hemolymph or the blood of the host? And when we look at the essential amino acids in the hemolymph of the ants in which we gave them back the antibiotics, so there's no bacteria present, or when we left their bacterial communities intact but did not enrich their diet, they look almost identical, right? We see no difference between the amount of those essential amino acids in their hemolymph. But when we leave their bacterial communities intact and enrich their diets, what we see is a huge increase in every single one of those essential amino acids in the hemolymph of the ant host. So in this case, what we know is that the bacteria within the guts of these turtle ants are recycling the nitrogenous waste of their host, transforming it into usable forms of essential amino acids that the ant host can use. And so since these ants feed only on plants, they're relying on their gut bacteria to provide the missing nitrogen in their diets. So Typically, I continue on this study, but I wanted to be mindful of time and also try to speak slow. For me, very difficult. But we recently published a study where we were able to demonstrate that the bacteria within the gut that are they're synthesizing these amino acids are then used by the ant host to build a hard cuticular armor. The paper's published, or if you want to talk to me about it, I'd be happy to talk to you about that. So hopefully today I've been able to convince you that using an integrative approach, using the lens of anti-evolution, but borrowing from whatever tools necessary to ask questions about what's facilitated this deep evolutionary relationships with plants, we've begun to sort of unsolve some of the puzzle. Particularly today, I've shown you that ants and plants have evolved this interdependence on each other through time, that in the case of these obligate symbiotic and relationships, it can evolve in predictable ways, not the way we initially predicted, but in predictable ways. And that lastly, the symbiotic bacteria have facilitated ants to shift onto these herbivorous diets and likely facilitated their movement up into these flowering plant canopies. So I really want to take a moment and thank my collaborators on the work I've presented today. I actually really want to thank my lab group. I get to work with the most brilliant group of scientists every day that are just so passionate and fun. Of course, I have to thank my funding sources, and most importantly, muchas gracias. Questions, please.
Thank you very much. I'm very excited with your study. Fascinating project. My very first questions were related with these evolutionary traits that can be linked with these changes in herbivory in terms of morphology. I was also curious about the mandibles and a lot of morphological traits. I was wondering if you could explain a little bit if it's also a restriction in terms of why it's not coming back with a more general.
That's a great question. So mandibles are used for lots of things, right? So of course for capturing prey, but also deterring predators. So we do see some herbivorous species still maintain big mandibles. But what we actually do see is that there's these muscles in the mouth that are related to sucking. So ants that are predatory are capturing things and stinging them and feeding them their to young. Things that are herbivorous are often out licking food sources, right, from extraflowonectaries. So we see that the muscles there have expanded. The other thing that I've never quantified, but I've actually always wanted to, is if you think about the digestive tract of things that are relying entirely on plants. So if you think about ungulates, right, we see that whole parts of their digestive tract have to become elongated so they have time to sort of pull the resources out. In all of my dissections of ants, I've noticed that predatory ants have very short iliums and herbivores have very long iliums. And so I'd love to actually go and have a student quantify that, because I think that there are morphological traits that are contingent with the change to the diet, which might be why we never see those reversals. But that's a great question.
Thank you.
Have one back there.
Hi, Corey. It was great. Thank you. So I have some our systematic students here. So some of my questions might be devil's advocate. I hope you understand. So you mentioned at the beginning that you use phylogenies because they answer a lot of how questions and why questions. I have always found that phylogeny might answer them, but sort of in a very unsatisfactory way. What do you think of that?
I guess it depends on what satisfies you, right?
That's true.
Ultimately, as an evolutionary biologist, I would love to have a time machine, right? So I could actually see when these things happen and why. We don't have a time machine yet. So we're left with looking at patterns and trying to then infer processes. I think that that's completely okay. Sometimes what we do is we can have windows into the process, the how, by sort of going at it in, non-intuitive ways or non-direct ways. And so I think that that's completely okay. And so some of those might be in a comparative methods way, right? So let's imagine that we think spines are an anti-predator defense. Well, that's nice. We can ask the question, how many times do spines evolve? But then we can ask the question ecologically, like, is that species more likely to be preyed on or not, right? So it can sort of use this, double-pronged approach to try to get to the answer. But for some people, incredibly unsatisfying, sometimes even for me, unsatisfying, but I don't mind pattern over just process.
Awesome. Thank you. So my other question, you mentioned that the earliest ants were predatory. And this is another devil advocate's question. How do we know that?
Yeah, so if we look at the aculeates, which are, within the Hymenoptera, all the earliest lineages, the wasps, that you think of are all predatory. The sister group to ants is the aculeates, which also contains, of course, the bees, but many of the speckled wasps, which are earlier in that lineage. And the sister to that is also predatory. Even within the ants themselves, all the earliest lineages of ants are also predatory. So really what we're sort of assuming is that we haven't seen a state change to this change in diets until further along, more derived in the ant phylogeny and more derived in the Apoidea phylogeny, right?
So that's an inference?
It is in fact an inference. Because again, I don't have a time machine to go.
No, but you have fossils. I was wondering if in the fossils you see something that points you to that conclusion that they were predatory.
Yeah, so in fact, all of the stem ant lineages appear to be predatory. Now, the reason I say appear is that some of them kind of look like every other ant. We can't be sure that they're predatory. There are whole lineages of bizarre ants that have all kinds of crazy mandibles for catching prey items. Most ants have mandibles that move like this. Some are like this. They have whole species that have mandibles that move like this with a basket on their head that pierce the prey through. So the fossil record also suggests that they were very weird predators.
Okay. Okay, I have another one. Sort of similar to Patty's. So sort of the transition is predatory, omnivorous, and then herbivorous, right?
Yeah.
Herbivory sounds like a trap because you are reducing your food source. I would think that omnivorous would be a more flexible strategy, right?
Yes. So, I think that the reason we see shifts in trait evolution within a lineage are often to take advantage of a novel resource or get away from competition. And so any of you who've, worked anywhere in the world, there's just a gazillion ants everywhere. How do you pack a gazillion ants into one habitat? Well, you have to take advantage of novel resources, but you have to reduce competition. So I think in many ways, it's a mechanism for reducing competition. I love your idea of a trap because we actually have in some species that ants that get locked into these are trapped by their host plant. The acacia ants are a classic example. And so, it begets the question of the chicken or the egg, which happened first. But I think that there are some advantages to being a generalist, right? Those are where we see almost all of our invasive ants coming from because they can adapt to any habitat. But it also means that you sort of are excluded from some microhabitats once you have a specialist. So I don't know that there's a satisfying question there, but I think that, I guess my argument would be that's how you pack 1 million ants into every habitat on the planet.
Right. Okay. One more. So yeah, they are both getting benefits in this mutualistic association, but the very aggressive ants, you will think they limit the opportunities from pollinators for plants, right? Because they are scaring off a bunch of other arthropods that might be pollinating. So the plant is paying a cost too. Absolutely.
And I would say that in those obligate mutualisms, that's probably more of a scenario. Interestingly, most plants have a that have relationships with ants, have these facultative relationships, right? So through things extrafloral nectars. Extrafloral nectaries are interesting because they attract any ant, whether the ant will defend or not. Most ants are actually wimps. They are not going to keep herbivores away. But there's been a whole bunch of ecological manipulative studies that have demonstrated that regardless of whether the ant will defend or not, having an ant or ants present on a plant means the herbivore would just rather move to another plant. So, I think that in most cases, you have a scenario where the ant is not tied into the relationship. They're only sometimes on the plant. So pollinators are able to access those plants.
Okay. I thought I have one more, sorry.
I think the crowd is going to revolt.
Really the last one. So what happened to the other bacteria that were in the crop?
So the bacteria in the crop are likely just contaminants. So I think that if we were able to sample the same individual ant, which we can't, through time, every time after they ate something, the bacteria in that compartment would just look like what they ate last. There's no sort of stability in that bacterial community in the crop. Yeah. All right, questions from others, or are we?
Thank you.
Okay, great. Hi.
Hi. So I'm going to take a little bit of what you guys were discussing. From a macroevolutionary point of view, you say there is a lock, there is a trap where ants can get into this relationship. That seems to be a lot like Dolo's law. But I think it's the thing is there's not enough time elapsed yet to see to escape to that trap, I think, because we've seen a lot of examples in very old lineages where you have that, but then you have species coming out of caves, for example. So don't you think it's too early to say that it's a trap? It may just,
what's too early?
We need more time.
I mean, so one of the things that we see in the acacia ant lineages, in that scenario, when I say it's a trap, the plant produces a type of sugar that's not common in most plants. And what they do is they change the physiology of the ant, the queens and the workers. So initially they're able to digest any sugar, but then because they're only provided with one, they soon shut down all those other pathways and now only can drink the sugar from this one plant. So even if you gave them other sugars from other plants, they can't digest them properly. So that's what I meant by a lock. And as in regards to time, I mean, these lineages are probably on the order of about 5 to 6 million years old in the acacia lineages. Maybe that's enough time. Maybe they'll still escape it. We haven't seen any reversals in any of the 3 mutualist lineages towards not being mutualists. It doesn't mean it won't happen. That's a great question though. And right next to you.
Hi, thank you. I work with legumes, and I can manage to find out how can we avoid that legumes, they can have ants living with them, but there are others that they use the ants just as scarecrows, right?
Yeah.
And that they don't only provide sugars on their nectars, they provide amino acids. The Baltian bodies are proteins, and they also have this symbiosis with Rhizobium and other genera of bacteria. So it seems that, have you tried to manage to make a study about the phylogeny of legumes and the phylogeny of ants?
That's a great question. There's a lot in that question, actually. First, I'll start at the end. So a phylogeny of ants and the phylogeny of legumes, I don't know how intimately I'd expect those relationships to be. I think it's pretty facultative. So I think that legumes will use ants when it's beneficial to them, but they don't always need, some species, but not all. The other part, back to the fact that in the acacia ants, they're eating these Beltian bodies, which are protein rich. We've actually done the microbiome of the acacia ants, and although they do have some bacteria that appear to upregulate their host nutrition, it's nowhere near the turtle ants. And because the turtle ants are essentially only tending sap-sucking insects or going to extra-flow nectaries, I think that the acacia ants have more in their diet, so they're not as reliant on their gut microbiome. And if there was something else in there that I missed, I'm sorry, we can talk about it at the coffee.
Bueno, si no hay más preguntas, podemos reunir con la ponente con café y galletas aquí afuera.
Gracias.